Hey everybody, in this video we are going to talk about initiation of DNA replication in eukaryotes. Now, many of you probably remember from maybe genetics or BSE 197 that DNA replication in prokaryotes, prokaryotes typically have one circular chromosome, and DNA replication usually starts from a single origin of replication, like that. And it usually occurs bidirectionally, right? So from the origin of replication, it'll occur in both directions, and we call this bidirectional replication. Now, eukaryotes, most eukaryotes have many origins of replication. So, and we know the most about DNA replication in yeast, so Saccharomyces cerevisiae. Now, in yeast, scientists have done a pretty good job at identifying the sequences that are found at origins of replication. We still don't know everything there is what determines an origin of replication. But in yeast, they are called ARSs for autonomous replicating sequences. And if you're curious, well, this name comes from how they were discovered. They were put into plasmids, these little sequences that were put into plasmids. They're about 100 to 150 base pairs long. And when they're in a plasmid, or a circular piece of DNA, and that circular piece of DNA is placed into yeast, well, that circular piece of DNA will be replicated if it has an ARS, because the ARS serves as the origin of replication. Now again, these are 100 to 150 base pairs long. And what I want you to do is take a look in the notes. There's a little more information about these. Essentially, they're A and T rich, so they have lots of A and T nucleotides. There's a couple of sequence motifs in them that seem to be important, but there's also something we don't quite understand. If they don't work at every location in the yeast genome. So this is consistent with what we know about, or what we don't know about, origins of replication in higher eukaryotes like humans. We know that they tend to be AT rich. And we think that it has to do, there is a component of a 3D structure. So how the chromatin bends and how it's arranged in the genome that makes an origin function as an origin. But for the most part, we still don't really know how they work in yeast, or what makes an origin in yeast, and what makes an origin in higher eukaryotes like humans. So I want you to check out the notes. It's just 1 or 2 pages of notes on these for some more background information. What we're really going to focus on in this video is the proteins involved in initiation of DNA replication in eukaryotes. So okay, so what we're going to focus in on mostly is initiation of DNA replication at origins in eukaryotes, and we're going to look at the proteins involved in this process. So let me diagram a long piece of DNA here. And we're going to focus in on just this section right here, which, let's say, I think this is should be about 30 base pairs. Now, at this location, if this is the general location of an origin in a eukaryote like yeast or human, now the first thing that's going to happen at the origin to initiate DNA replication, the first thing that needs to happen is we need to have these 6 different proteins recognize the sequences at the origin and bind to them. So these proteins are called ORC1 through ORC6. So 6 proteins in total. And when they bind, well, that's called the origin recognition complex, or ORC, origin recognition complex. So after the origin recognition complex forms, 2 proteins come in, and these are called the licensing factors. And they are CDC6, and CDT1. And they bind to the ORC. And they are called the licensing factors. And they are important because the process needs to occur, initiation of DNA replication needs to occur at each origin once and only once, each cell cycle. Because you can imagine if it occurred more than one time, well, then we would have certain sections of the genome being replicated more than once, and we'd have duplications. And that's generally a bad thing for a genome. So they are the licensing factors, and I'm going to talk a little bit about how they function as licensing factors to make sure replication only happens one time. I'm going to talk more about that in a moment. So after CDC6 and CDT1 bind, well, they can help load on the MCM complexes. So these are made up of MCM proteins 2 to 7. So that's 6 proteins in total on each side. So here we'll have 6 MCM proteins, 2 through 7 over here, and 6 over here. And what these are the helicases. So these are the helicases that will unwind the DNA during replication. And remember, we said DNA replication occurs bidirectionally. Well, this one will go that way with the replication fork that moves to the left. And this one over here will go this way. Of course, these things will have to be moved out of the way. Together, all of these, this MCM complex here, this MCM complex here, CDC6, CDT1, and the ORC, they're called the pre-replication complex. So this whole thing is the pre-replication complex. Now, in the next phase, what's going to happen is CDC6 will be degraded after the MCMs are loaded. CDC6 can be degraded. And then another protein called geminin, what it can do is it can bind to CDT1 and inactivate it. So even if these helicases, if replication was initiated, even after these took off, well, there'd be no more CD6 around, and then any CDT1 that was around would be inhibited. This replication origin could not be licensed again to initiate replication again in the same cell cycle. So this origin is done. It can't be licensed again because CDC6 is degraded, and the cell won't make any more CDC6 until it goes through a complete cell cycle, until the G1 phase starts again. So that's kind of how the licensing factors work. So they can load one set of MCM complexes on, then after that they're going to be inhibited. Okay, so in the next phase, I think we're out of order here. Yes. So it seems like it got really complicated all of a sudden, but no, it didn't. Don't worry too much about that. It didn't get too complicated. We're going to add 3 things here. So MCM10, GINS, the GINS complex, which is a complex of 4 proteins in here, and CDC45. So we're going to add 1 of each of those to this MCM complex here, and 1 of each to this MCM complex over here. Now this other protein, this is a kinase, and it's thought to be important for helping CDC45 load onto here. And what do all these proteins do? If you want to see more of what they do, you can check out the notes. But for the most part, we're just trying to, take a look at all the different pieces that are involved in initiating replication. So when all of these are on here, what we're going to call this, the CMG complex. And some people call it the CMG helicase complex. The C is for CDC45, the M is for MCM10, and the G is for GINS. So each of these would be a CMG helicase complex now. Then we have a little factoid down here that MCM10 is thought to chaperone the polymerases that we'll have to come on to this next or in a subsequent step. And the final thing we're going to look at when it comes to initiation of DNA replication is, again, in the previous page there, we said DDK, or the DBF4 dependent kinase, DDK, was important for loading CDC45. Now, it's also important for phosphorylating a number of different proteins involved in within the CMG helicase complex. So most importantly, proteins of the MCM complex here, the MCM proteins in here. It will phosphorylate those, and the phosphorylation of the amino acids in these helicases are thought to be important for activating the helicase activity and initiating replication. So lots of other things are going to have to come in here, right? We didn't mention the polymerases, right? There are 3 different polymerases that are going to need to come on here. And so the next step with all of these other replication proteins, when those come on, we're going to call that the eukaryotic replisome. So that's it. That's all we're going to talk about for DNA replication. What we're going to take a look at next is, how do scientists know, these are the components that assemble at the origins to initiate replication. We'll take a look at how they know that and how they do studies of these complexes. We'll take a look at that in the next video.